So a very good afternoon to everyone, and thanks IOB and IIT to put this wonderful workshop and the conference that starts tomorrow. I'm really looking forward to various talks that are gonna be there, and I've been on and off since morning listening to various speakers of previous like from the previous levels today, and I'm pretty sure that all of us have learned a lot from them. So as a follow-up of how do we basically do reproducible science, as Abhishek was referring to, the effort for our initiative here, like now and next 45 minutes, and then after again 45 minutes is to introduce you to a workflow system called Galaxy. Okay, and we'll learn during the talk why do we need to do this, how it can be of real help, and what are our efforts to ensure that we have a wider infrastructure that can be made available for the Indian scientific community to make reproducible workflow systems. Now, just to start off, the title of my talk says Introduction to Galaxy. Now, if you go and type Galaxy as a keyword in Google Images, this is what you get. So you really don't get anything more than this. You get more and more pictures of Galaxy. So if you are looking at Galaxy workflow, you should write Galaxy workflow and you start seeing images like this. Yeah, for people who actually have worked with Galaxy before, it makes sense, but the newcomers out there may need to know more about what's going on here. So what I'll do is I'll simplify the task for the new people on board who want to understand what workflow systems are. So to put it in a very, very simple words, what is a workflow? Now, if you see here, it is a conveyor belt, and on a conveyor belt there is a pizza dough, and as the pizza dough moves forward on the belt, a particular pan is adding a specific amount of sauce, the next one is adding a specific amount of cheese, and the last one is adding a specific amount of mushroom. Now this workflow clearly indicates that every time a pizza is coming out of this workflow system, the quantity and the amount and the way it is being thrown at the dough is the same. So your expectation would be that the taste of the pizza at the end of the exercise is going to be reproducible. It's going to be exactly the same. Now, the minute I replace these saucepans with something which is more manually done, you know there's going to be variation, right? And the whole point is that these variations in pizza may not impact us so much, but if we have these variations in the way we do data analysis, it's actually gonna mean something completely different at the end of the work. So in most of our cases, this is how a workflow looks like. So we actually don't work with simpler problems. We have several steps, irrespective of whether doing a computational experiment, whether we are doing a wet lab experiment, there are several steps to it, and there are several decision points, right? Now assume that I have executed a huge project and I have these so many steps where I took decisions, right? At some point, I don't want to basically do this, right? Now, if I change this step and I have done each one of these step manually, what's going to happen is everything that comes afterwards, I'll have to repeat the whole thing. Okay, if I make a small change here, things upstream and downstream, I need to change this if I continue to do and run individual applications in a manual manner. This is what Dr. Abhishek was also referring to at his lecture when he was teaching you Python, that in a programming concept, everything has to be streamlined. You change one thing and the rest also needs to align to it accordingly. So having said that, your workflow design can have several challenges, and to overcome these challenges, what you have are workflow systems. So a typical workflow system is basically the main objective is to perform routine, extensive, complicated tasks. And if I replace it from making a pizza to data analysis, it can mean anything from data transformation to data mining to data analysis. So point is, it is a repetitive exercise done by a system. So that will again means, because if there is no manual interference, there is reproducibility. Every time you expect to get the similar result, irrespective of who is doing it. You can share these protocols, so the details of the methods are shared with somebody else exactly how you did it, and not by writing it in materials and methods in a paper that can sometimes miss a very critical point and render the whole exercise non-reproducible. transparency in the protocol. So again, the point, if I have done some analysis and I want a field expert to review it, I can actually send the entire workflow, the analysis pipeline as is. Somebody can look at it and say, okay, at step 8, instead of 0.8, you might like to put up a value of 0.1. And I don't have to do everything again. Then yeah, just rerun the workflow. So it's giving me advantages of reproducibility. It is giving me advantage of sharing, transparency, and it is basically a community-centered exercise, because not, as you can see, with more and more interdisciplinarity, as your previous speaker was also referring to, the minute the interdisciplinarity of a subject increases, it actually indicates to the point that many collaborators start working on the same project, which means that you need to have transparency in protocol and understanding of what's going on with each of the partners. Now, to give you an idea, let me just go back and see if there are any comments on. Not so far. All right, so we are good. Okay. So now this is basically a cartoon showing that we are actually drowning in the big data. And I'm not talking of big data as Harpreet was referring to from many perspectives. I'm just focusing on genomics. And if you see, the genomics data projection is at 1 zettabyte basis per year by 2025, which is not far away. Which practically means that if we need to make any sense of this data, we need to have workflow systems that allow us to churn information from this data in a reproducible fashion. And as already highlighted by previous speakers, in addition to workflow systems, or you want to use workflow systems, there are several components on top of it that one has to learn today. You must learn data analysis, which includes machine learning, AI, network science, predictive modeling. There are several domains that one should know today, and workflow systems try and help you put it together in a simpler fashion. If you have developed something and it has to be used by somebody who is not an expert in the field, a workflow system allows them to be used by a wider community. And this is again Minion from bench to bedside. So the point is your sequencing machine practically looks like a pen drive right now. So the data size that we are talking about is only going to increase in future. And with increasing data size, the skills in data analysis is what is the current and the future market is going to be. And of course, people who are doing data science will also be need, you are, as data scientists, we are required to provide our applications and platforms in a way that somebody else can use them without learning too much of everything, like a mobile phone. So somebody who developed a mobile phone, we don't know how it is developed. We use it, right? We still can use an application which we cannot build on our own. But data analysis protocols today are very difficult for somebody who is not from a computational background. So we need to provide these workflow systems in an easy to use fashion by a wider community. So that brings us to Galaxy, and what I'm showing here is actually applicable to any kind of a pipeline based or a workflow system. So a simple thing is that you have a question in mind, and with that question you start with an input. Like say for example, I have sequenced a new genome and I would like to now know how many proteins that sequence has so that I know the functionality or the capacity of that genome. So say, okay, this is the kind of function this genome can do. So that genome file becomes the input. My job one becomes, say for example, doing the structural annotation of the genome. So I can use gene scanner. I can use several other methods to really identify how many ORFs are there. Then those ORFs becomes the output one because they're output of job one, but they become the second input for job two. With job two means now I know that these are the fragments which may code for an ORF, and now I need to functionally annotate them. So my job one becomes structural annotation, my job two becomes functional annotation. Once I have done the functional annotation, I would like to do a pathway alignment. Once I have pathways, I need to do more beyond it. So depending on what my question is, I can keep on adding the number of steps to a workflow system, provided what my question is. So I know my question, I know my data, and then I'm trying to do a stepwise analysis of that data and building my workflow system. Now, as you can very well realize that the question that you are asking versus what I am asking may differ. So we need to basically mix and match these jobs in a way that suits our requirement. And that is why any workflow system which has to be used widely should be modular. I should be able to play with the components, and it should not be a rigid structure which says, okay, this is what you have, these are the steps you need to follow and no modifications. That won't work. because every question is unique, and every question needs to be addressed in a very specific manner. We can still use the same combination of tools, but in a different fashion. So that gives us, an infinite choice of combinations in which we can run this. So in order for us to understand what I'm talking about, I have taken a small example. So for a very simple understanding, if you see here, this is nothing but a eukaryotic cell, and we know that we all have a nucleus which has a DNA, but we also know that there is something which is called as mitochondria. And mitochondria, there are several in a single cell, and each mitochondria, when you bloat it, you will see that it also has its own DNA. So it's a semi-autonomous organelle. Though the DNA is small, it actually encodes for 13 protein coding genes, 22 tRNAs, 2 rRNAs, and some non-coding segment is also there. But more than 3,000 proteins that are required for mitochondrial function are encoded by nuclear DNA. So the point is, what you have here is a circular DNA, which looks like a bacterial origin DNA, because the nuclear DNA is not circular. So the point is, you have this small DNA, and the reason I am focusing on this DNA is the following. Mitochondria is responsible for several diseases. And if you see this graph, we did a representation a few years ago in our paper. We have showed that in past 15 years, the application of mitochondrial DNA sequencing in medical research, as you can see by the increasing width of this green bar, is increasing over time. Why is it happening? One of the simpler thing is this. Though there are a very large number of tests that are available to diagnose mitochondrial diseases, they are not affirmative to 100% value, in the sense that just by one test you cannot diagnose it. That is why the Mitochondrial Medicine Society has recommended next generation sequencing as the first line of diagnosis for mitochondrial diseases, which means that when a patient comes and the doctor feels, based on clinical symptoms, that this person can have a mitochondrial dysfunction, the first step is to sequence the mitochondrial DNA and not the last step. So it actually has a direct clinical application. So now in this context, where does a workflow fit in, and why do we really need it? So if I can take you to the next slide, you can see that this again is a circular representation of a mitochondrial DNA, and the only difference is this time I have mapped all the genes and a few tRNAs that mitochondrial DNA encodes for. And on the outside, I've listed all the clinical diseases that mitochondrial DNA has been shown to be responsible for. The issue is that there is a huge overlap in terms of symptoms of these diseases. So one-to-one correlation, like if you see here, a small section, there are so many diseases caused by a very small number of tRNAs. And sometimes even a single modification sometimes can lead to several diseases. So there is a genotype to phenotype complexity that even after sequencing is hard to resolve. And given that it's not just ATP, mitochondria is involved in several functions, you so see this heterogeneity of symptoms. So the point is now all of this data, the sequencing data and the clinical data, has to be put together to make some sense and to do an accurate diagnosis. So my lab has been developing several algorithms for past more than 12 years to basically diagnose and understand how a patient mitochondrial DNA can be used to identify prioritized set of pathogenic variants. So this is one of our first results, which we called as MITSNP score. It's a pipeline. And at that time, if you go to the original paper, this was not built in Galaxy. This was built like a routine web server. So the point is that for ataxia or Alzheimer's disease or Parkinson's disease, obesity, we could show that despite the mitochondrial DNA has very large number of variation, our pipeline can identify a very small set of variation that can actually be pathogenic. And then we need to supplement this information with respect to different population groups. So we developed MitoLSTP, which is nothing but a repository of more than 5,000 individuals coming from several ethnic groups in the world, because we know that a specific mutation background has an impact on the phenotype. And then we also developed visualization using a browser. The problem is Despite the fact there are three different applications that even developed only in my lab, I actually find it a nightmare to link all of them together. Forget about linking all the resources that are developed in the mitochondrial community so far. So even the applications that I am developing, I have the source code, I have all the know-how. Despite the fact moving from one application to the other that we have developed was extremely hard. And that is when we actually made a decision to go ahead with integrating all the applications that we have developed. So basically putting the data together, which can be searched using a query builder for mitochondrial mutations or for the proteins encoded by nuclear DNA, to variation analysis, which is not just the application developed in my lab, but also other tools which are developed by other people, other groups. And how do we visualize data? And of course, you will see and learn from both Rakesh and Tina downstream that how the applications cannot work in a similar fashion and can lead to different set of results. So you need converters and mappers. We'll come to that. So this is basically a workflow system. This is how Galaxy's interface look like. So what you have on the left-hand panel is the tool list, all the tools that you have. In the center is a canvas, and on the right-hand side is a history. So exactly when I told you job one, output, input one, job one, output one, input two, job two, output two. This 14, 15, 16, 17, 18 is exactly representing the same thing. These are basically outputs of individual jobs that you have executed, and you can see them on the canvas. So what MitoLink currently has, it also has a library, which is coming from 5,000 whole genome sequence data sets, over 600 populations, 27 different phenotypes, and more than 130,000 variants. And all of this is now available, so anybody can actually now ask different questions. Like say, for example, as you can see on the screen, I have shown data on diabetes. Now, I told you that there is a lot of overlap in different mitochondrial diseases. So now if I would like to basically segregate or understand what are the differences between neurodegenerative diseases like Alzheimer's or Parkinson's versus metabolic diseases like diabetes or obesity, how do I segregate the two? So I can actually ask these questions using a workflow system saying prioritize mutations for diabetes, prioritize mutations for Alzheimer's, show me the overlap, which gene they belong to, and can I now visualize them on a browser? So all these questions can now be asked, and we are not limiting anybody to ask any question on this data. So which makes it easier for everybody to reuse and rebuild several types of analysis workflows. And again, it gives you a very large number of information, and more than 3,000 nuclear DNA proteins with mitochondrial function are also available. So the point is, despite the fact that my lab published so much of mitochondrial DNA work in past 12 years, till the time we put all of that together in a single workflow system, which is MitoLink, it becomes very hard for it to be used for any clinical application because the data was available like at 10 different places, and you cannot expect a clinical setup to actually do this analysis going to separate windows. And because it's a hard decision for patient therapeutic intervention that you need to have everything at one place. So now we are actually, so I'm working with the Mitochondrial Disease Sequence Data Resource Consortia. It's a global consortia of majority clinicians who are who basically see mitochondrial DNA patients on a daily basis. And we are trying to develop MitoLink as a platform where the clinical data also comes into picture, because we know that we cannot release clinical data out without compromising on patient data security. So the point is we are now developing MitoLink next level to make it accessible for clinicians. So once they sequence mitochondrial DNA from a patient, they can import it here and do any kind of workflow system development to basically have clinical applications of what we developed in the lab. So this is basically a simpler overview. Subsequent to MitoLink, we also parallelly have been developing other workflow systems. This one is particularly for people who would like to work with drug discovery. This is called Molecular Property Diagnostic Suite, and as you can see, it has several modules. I'll not go into the details of this. Rakesh and Tina will, after our session is over with me and Harpreet, they will tell you how to install Galaxy, how to integrate tools, how to basically integrate workflows. And Tina is going to take a specific example of how do you start from nanopore raw data all the way to identify a phenotype of antimicrobial resistance. So she's going to explain you that in much more details. The job here that I'm doing is to tell you what our lab does in context of the mitochondrial diseases and not so much the infectious diseases. The other thing, don't get confused by too many things written here. The whole point is that you can integrate applications, you can run those applications, and you can actually write APIs to migrate and show these applications on lab notebooks. So you can actually convert the entire protocol into a workflow system, which is like a computer program that also can be shared. So this is basically trying to say that maybe there are 2 labs or 3 labs that are generating these resources, but since Galaxy can be made available on cloud, they can be accessible by a very wide range of research community. And because they can actually, share and reproduce and redo what has happened, this actually is one of the first, like a very strongly recommended platform for reproducible science. So I would recommend as many people to use it. Now, this is basically a timeline of projects what I have handled in my career so far. And if you see here, Galaxy will appear at several places as early as in 2010 when the Galaxy community just began. And this is when we started using Galaxy for drug discovery projects and subsequently Galaxy for several other aspects. This is Galaxy for MitoLink, and there are other things with Galaxy we have done as well. So, but I'll not take you through everything. It is to show you that time and again, we have been revisiting Galaxy because they are, as a community, I'm a member of the community. The Galaxy core community actually keeps on adding more modules and tools and making it even more exciting for us to see how do we apply it in our projects. Now, galaxyproject.org is the place where you go and look at all the resources that Galaxy has to offer. And as you can see, there are use Galaxy resources, public servers, commercial clouds, containers. So you have a lot of information on Galaxy that you need to read. 20 minutes is not the time to really cover all of that, but I need to give you pointers so that you can follow up. And as you can see here that we have Galaxy in EU, we have main Galaxy, which is from the Penn State and other places, and then you have Galaxy Australia. And these are public servers, and as you can see, one of the first public servers listed here is AB Open Lab, which I showed you in my previous slide. So whenever we publish something in Galaxy, Galaxy centrally offers us to basically advertise them on their server. So you get wider visibility of the tools that you develop here. So a strong recommendation, build your tools, import them in Galaxy, put it out there. A very, very vast community will be able to access them. So this is a slide that I wanted to share with all of you, and Harpreet will also join me in explaining this. So the point is that we were, both of us were asked to share our plans on Galaxy India. So we thought if there is a Galaxy Europe, if there is Galaxy Australia, can we think of Galaxy India and make it like a more centralized workflow system for our scientific community? And we presented a few ideas that we have during this Birds of Feather meeting, which happened 2 months ago. So this is basically what we are planning to do. We have Harpreet, who's going to tell you about BioClues and how we are basically harnessing the network that BioClues has already developed to set up the Galaxy community in India. And we have Prashant, who has been leading BioClues since the very beginning. He's a founder. And we have international advisors like Sonika, Kiran, and Prateek, who have developed immense number of applications in Galaxy, and they teach it to the rest of the world, and none of them are based out of India. Their faculties are all across the world. So we actually have some loosely built structure in place as of now. And the plan is, can we have, if Galaxy India has to materialize, we need a few things. First of all, we need to immediately start. So we were thinking we can use the CSIR resources, the CSIR MTech resources to already launch the Galaxy India project. Of course, the Galaxy also, like as I have shown you, we have research projects hosted on Galaxy already, which are published. So I don't see a problem there. But of course, it needs to scale up for infrastructure if we need to provide it as a service to a wider scientific community. Then we need to have partnerships. So BioClues has come forward in a big way, and Harpreet will tell you more about it as we go forward. Then we need to have outreach, training, and workshops, because as this workshop is organized by Abhishek very, very proactively, I think this is an outreach activity where we are informing the community of that about the fact that we are interested in launching Galaxy India. And of course, we need funding to make it sustainable. So just a very, very quick pointers before I hand it over to Harpreet. We wrote, so Dr. Prateek Jagtap, he's at University of Minnesota, and MTech and Prateek wrote a joint proposal into US Science and Technology Forum, and we have funding to basically organize Galaxy specific workshops in India. Because of the pandemic, the plans are a little dwindling, but this will happen very, very soon. If not physically, we'll do an online event which is heavily focused on Galaxy, but revolving around specific biological problems. Then we were also thinking of riding on the Jigyasa program that CSIR already has, where we have interfaced with a couple of thousand school kids across the country. We can actually use Galaxy for education modules, and as you know, we have 4 to 6 month training program in India. We can customize it them so that the students actually learn Galaxy and become ambassadors of the community and make it more widely applicable. And when I say this, believe me, Galaxy is one platform that we can use because of the ease of how to set it up, and you will learn that in next 20 minutes, how easy it is to set up and how easy it is to integrate workflows and tools. But of course, for reproducible science, there are other workflow systems already also available. I have used a few of them, and after doing all that exercise, I still would recommend Galaxy. But if you have more ideas on introducing other workflow systems, I think that is something as a community we should discuss. And we should organize thematic workshops, as I told you initially that we are doing a few. This one is also one of them. And how are we leveraging on BioClues community is BioClues is something that Harpreet will tell you about. The only idea is that we are trying to use BioClues network for operational purposes and also to see if they can launch awards for demonstrating reproducible workflows. That is a way of incentivizing the community in the right direction. We have the National Knowledge Network. We can partner with them for dedicated infrastructure. This is something which is in discussion right now. We don't have answers because this has to be done through a proper channel of making it really functional. And why we recommended MTech? Because of several reasons. We are currently number 4 among all institutes in the world who are providing biological specific data sets to the community. But as you know, Galaxy can be used for anything. Of course, it's a biology heavy community, but of course it can be developed for other applications as well. And this is just for selling my institute or telling you more about it. We are the Informatics and Big Data Analytics unit at MTech. We work on several aspects. Our servers get several million hits per month from many institutes across the world. And we do organize trainings and workshops, and we're very, very happy to do more and more workshops focusing on Galaxy as the central point. And since we have interfaced to a wider student community, and I think Harpreet has interfaced to even wider student community, can really do a good job at popularizing it. And of course, this is again to show that we publish with very young kids, even when they are in school. So we engage with the scientific community in a very, very serious manner, not just for outreach, but we engage them so that they can learn how to do science. And this is a paper from my lab where the first author, Pooja happens to, when she published her paper, she was practically in her school. So it's not that we're doing only outreach. We want to do serious science. So, and that is what we thought, that if we have the capacity to do that, why not guide our younger generation towards doing reproducible work? And for that, we need to have platforms because of the sheer size of the data we are dealing with, the number of applications that come every day, the number of data formats that are out there. There has to be some consistency. Reproducibility requires that all of this information has to be made available in a systematic fashion. And yeah, we work with many students. These are all undergrad, grad students, school kids that have published with me over several years. This is just to show that I personally, the reason for me engaging with the Galaxy India program was the fact that I really like community engagement. And this is basically, before I say thank you, because I can understand there will be several students watching it on YouTube and as well as here. This is a drug discovery hackathon which is going on right now with the government of India to solve COVID-19 problem. It's specifically focused on drug discovery, but there are other things that you can do. Please go here and see if you can apply your brain in addressing the pandemic. And with that, I would really like to thank all the people who I have learned from so much. and would request Dr. Harpreet to take over and tell us about the BioClues organization and how BioClues is coming in a big way in basically, we are actually thinking of having Galaxy under the umbrella of BioClues, and Harpreet will tell us more about how do we plan to do that.
Thank you, Anshu. It's a very nice talk, and you set the stage by giving an overview of what workflows are and how workflows can be implemented in the big size. And today is the era of community, projects. We are solving very big problems, and so we need to know about the systems like Galaxy. And just for your knowledge, though entire world is working on community projects, in India still, except few labs and few, much aware scientists, most of the peoples are not aware of what is the power of community science, what is the power of doing working in big teams or even working in teams. Let me share my screen so that I can pinpoint on what we are doing here. So that's why, just to make people aware of community projects and particularly the work Galaxy is providing, we are trying to launch Galaxy India community. And as Anshu pinpointed, we had a very good discussion during our BCC 2020 conference, and it's very good that we got another opportunity very soon to discuss about Galaxy India plan. So before launching of community, you must understand that community launching is also a very big task, very, require much more manpower, much more thinking, much more networking. And luckily, in India, we have BioClues organization, which is, master in doing networking, and it's a very good community already developed in India. So what we are trying to do is we are trying to leverage the networking, the experience, the ethics of BioClues, just to make Galaxy India happen as soon as possible. Most of the people might be aware of BioClues organization, but I felt it's my duty to introduce BioClues organization to all the speakers and getting the opportunity of this platform. So BioClues organization, if it's a bioinformatics club for experimental scientists, and it was founded by Dr. Prashant Survajala and Pritish Vashwas in 2005. We have a present president, Dr. G.N. Shastri, Narhari Shastri. He's a director of CIR NEAST, and it's registered for non-profit organization, and we are very much adhere to the Creative Commons license. We have more than 4,300 members across 40 countries, and many of the students are have been mentored by BioClues for doing large number of projects. We have finished many projects, particularly 30 virtual projects, and involving around 2020 scientists from different research institutes across India, in fact, across world. We have a lot of collaboration. I will show you in detail when I move to my next slides. And we have been affiliated to International Society for Computational Biology, as well as the Asia Pacific Bioinformatics Network in India. Just to extend the core team, something about the core team, we have a team, dedicated team with secretary, joint secretaries, finance chairs, women biology chairs. In fact, we have really recently established a bioinformatics for school chair, and also a student chapters here. So we are trying to involve as many people as can to make BioClues reach the entire all the roots of this country, each and nook and corner of this country, so that people can take advantage of this growing science of bioinformatics and computational biology to make their projects, to take your project to a next level. We also have an honorary advisor, the great scientists who need no recognition: GPS Raghava, PK Gupta, Kavi Kishore, HS Balian, DPS Verma, and many of the others. So this is a journey we have launched in 2005. We start mentoring the students in 2006, We started offering fellowships to needy students in 2008. We started a women in biology chair so that the women folk of the country, the scientists, cannot be left behind. We also got affiliation with AP Bionet and ISCB. In 2010, we introduced the bird word. I will let you know what the bird words are. In 2011, we launched our own international journal for computational biology. In 2012, we launched our own cloud-based servers so that different data sets and server developed by BioClue's research and innovation cell, they can be put there for use by the general public. In 2016 onwards, we started introducing student chapters, and we also initiated somehow bioinformatics for school children, because we thought that the grassroots level, the school level is an ideal place, an ideal time for students to engage in science. 2019 till end, we had a very good partnership. Right now we are extending, or we are trying to build a very strong partnership with Galaxy community, so that the Galaxy BioClues joint community can be launched in India very soon. And we have a vision in up to 2020 to make bioinformatics well established in this country, and bioinformatics and BioClue is being playing a major role in all the major projects. So this is the mission, and based on four alphabets like more, mentoring, outreach, research through awareness, and entrepreneurship. If we talk about mentorship, so we are having more than 4,000 active members, and already many of these members have been involved in various research projects. We have a women in biology forum where we try to involve women biologists or women members of the society to do some good research. We have been generating many publications, around 25 publications in the recent years, and we are actually a thriving bio community. These are the new initiative as far as the mentoring is concerned. We have created BioClues interest groups, which are the WhatsApp groups, Facebook interest groups, so that different people working on different areas of science like genomics, plant biology, and computational biology, big data analytics, they can discuss their problems day to day with the mentors, and so that they can solve their problem, they can learn new things. And also we keep on posting the research and developments, recent research and developments happening in their respective areas, in their respective interest groups. Additionally, we have a career group for North chapter and South chapter. Also, we have the groups like microbiology, agribiopods, structural biology, and genomics. We recently introduced cyber session, keeping in view the COVID pandemic and keeping in view the importance of virtual events. So we try to have a weekly conversation of students and all other members with the mentors in BioClues, so that they can discuss their problems in detail, and they can be made aware of what is happening inside. Also, we launched it in 2016, but it we made it operational, and we revive it in 2020, the things called bioinformatics for schools. So bioinformatics school is right now launched in a pilot, as a pilot project, where around 20 students from different parts of the countries are learning bioinformatics, how bioinformatics can be used to solve different problems. Outreach, we are trying to align to the new normal. We already having already conducting many workshop and conferences, and the notably the INBIX series, the Indian Conference on Bioinformatics, is the hit for from BioClues Society. And we had planned to conduct the INBIX in May, but due to pandemic, this has been postponed indefinitely. uh, but we try to fill the gap. We in the pending time, we try to arrange many virtual conferences, many panel discussions on burning issues like secondary agriculture in India, COVID diagnostic, and functional genomics, and so on. So this is what we are trying to follow as a new normal. And we our recent discussions or recent conferences, upcoming conferences have been listed on the website right now. Research and development, we carry out research in different areas, including functional genomics, system biology, big data analytics, and we also encourage training and educational programs. We also try to make people aware of different employment opportunities through Women and Biology and Journal Club. We try to discuss various burning issues so that the new research can be initiated, and we are trying to make many people involved in joint research papers so that some of the new people, the novice one, they can have idea of how to write research papers, how to how to do a real biological problem, how to solve a real biological problem, and so on. We also, as a part of our research through awareness, we launched BioClue's Innovation Research and Development Bird Awards, and it's a procedure award which is given after a very thorough process for the scientists, working professionals, medical professionals, and other peoples working in the area of functional genomics and bioinformatics by nomination. Nomination are awarded each year, and in fact, the call for the recent nomination is open in September 2020. In 2019, we were not able to award the Bird Award because none of the participants they met the expectations at that time. This is a list of different persons who have been receiving the Bird Awards for the last 10 years, and some of them have reached many new heights, and they are heading the genome. For example, Vinod Sakaria, he has been instrumental in handling the genomic revolution in India from IGIB New Delhi. He was awarded Bird Award in 2014. So you can sense the importance of these awards, how critical these awards are, and how much work BioColleges does in recognizing people who are doing real work in the area of bioinformatics. We have been conducting many projects, and these are some of the projects which have been mentioned here in collaboration with the Cancer Institute Chennai, Gudvasu University Ludhiana, and so on. We have been expertise in many areas, but NGS and system biology are the main theme in which we are working very much. So this is an NGS pipeline which we have been developed in our labs, and we are trying to make people use that pipeline. So now we are trying to use Galaxy workflows so that these pipelines can be extended, the use of this pipeline can be extended. Even the peoples, the students, and the mentors working in the colleges and the remote parts of the country, they can also be make aware of how this big data created through NGS experiments can be used to ask very specific questions, and how that question can be solved using big data analytics and the workflows available in Galaxy. These are the services which we provide: mentoring graduate students in bioinformatics. We annotate and curate NGS data sets. We do transchromic and genomic analysis of NGS data, pathway analysis, and so on. I think there is no area in bioinformatics which is not been touched by the mentors of the Bioequilibrium Society. They have been expert in all the areas, leading ranging from the medical genetics, medical science to agriculture science, environment. They're working in all aspect of biology. How we sustain, this is something very important. We have a paid membership. It's very nominal one, only 300 as a student membership and 2000 for the live membership. We also have a $100 corporate memberships per year. We also try to generate some money through virtual and on-site conferences. We also try to earn some money from project sources, but that is only on the voluntarily basis. We also thrive on various grants and funding to support our endeavors. These are the collaborations: ISCB, AP Bionavit Technologies, Pine Bio, Nanotemper Technology, and recently we will also be joining hand with Galaxy community to launch Galaxy India. And with this much information on BioClues, I am thankful to organizers of this international conference of data science and biology, and especially Abhishek Kumar. And I'm also thankful to my co-speaker Anshu Bardwaj for planning this workshop in a very short period of time. Thank you very much. If you have any question, you can get back to us. Thank you.